Omeprazole. Contains introduction of omeprazole, medical uses, history, chemistry and stereochemistry, Esomeprazole, metabolism, mechanisms of inhibition, adverse effects, dosage form. Introduction. Omeprazole, first proton pumping inhibitor drug, inhibits the secretion of gastric acid by irreversible blocking enzyme system of hydrogen potassium ATPs in parochial cell. Racemic mixtures of 50% is omeprazole and 50% are omeprazole. One of World Health Organization list of essential medicines. Medical uses. Used in treatment and management of peptic ulcer, gastroesophageal reflux disease, heartburn. History. First marketed in United States by AstraZeneca under brand names Losec and Prilosec. In April 2001, AstraZeneca introduced Esomeprazole as replacement drug.
Chemistry and stereochemistry. Omeprazole contains a tricoordinate sulfonyl sulfur in a pyramidal structure. Have S chiral center, therefore can exist as either the S or R enantiomer. Omeprazole is a racemic equal mixture of two R and S. Omeprazole optically inactive, but esomeprazole is active. Esomeprazole. Makers of omeprazole were trying to find a drug with a better bioavailability.
Substitution was varied on both the pyridine and benzimidazole ranges.
This compound was eventually found to be the S enantiomer of omeprazole, esomeprazole.
Studies show that esomeprazole has longer duration of action than omeprazole.
Esomeprazole proved to be the drug of choice for management of gastroesophageal reflux disease. Omeprazole and esomeprazole. Omeprazole is R and S racemic mixture. Esomeprazole is S isomer pure. Omeprazole is less active. Esomeprazole is more active. Omeprazole is large dose. Esomeprazole is small dose. Omeprazole is a prodrug. Esomeprazole is active drug. Metabolism.
Occurs primarily in the liver by cytochrome B450 enzyme family, mostly by cytochrome B2C19 and cytochrome B3A4 enzymes.
Reactions: sulfone formation, hydroxylation, and orthodemethylation.
Omeprazole is substrate primary for cytochrome B2C19.
R enantiomer is cleared more rapidly by cytochrome B2C19, mostly through hydroxylation.
Hydroxylation is much slower in the S enantiomer.
It is thought that hydroxylation is enantioselective.
Combined effect: high bioavailability in case of esomeprazole.
Recent publications have been reported that this effect is due to the stereochemistry of the drugs.
The proton pump.
Gastric acid secretion occurs from the gastric parietal cells.
Parietal cells contain receptors for acetylcholine, histamine, and gastrin, which increase hydrogen chloride through second messenger interactions upon binding with their respective ligands.
In 1976, the proton pump was discovered, which contributed to hydrogen proton in the formation of gastric HCl.
It was postulated that activation of the proton pump through acetylcholine, histamine, and gastrin led to increased acid secretion.
The proton pump.
The proton pump is an ATP-dependent hydrogen potassium exchanger.
It is an enzyme that is integrated into the membrane of gastric parietal cell.
Quaternary protein structure made up of 1 alpha subunits and beta subunits.
Alpha subunit contains active binding site for ATP.
This site is the target binding site for protein pump inhibitors.
Beta subunit is involved in correct membrane integration and targeting of the enzymes complex to the parietal cell surface.
Upon binding of ATP, the protein pump undergoes conformational change known as E1 and E2 states.
This change allows exchange of one proton from the parietal cell into the gastric lumen for every potassium. Mechanism of inhibition.
Proton pump inhibitor carried through blood to the gastric parietal cells.
Proton pump inhibitors are weak bases and prodrugs.
Activations occurs via protonation at high acidic environment of the gastric lumen.
Leads to formation of tetracyclic sulfonamide structure.
Sulfur atom of the proton pump inhibitor binds to the sulfhydryl groups of a cysteine residue located on the active sites of the proton pump alpha subunit.
Forms a sulfur bridge through covalent bonding, causing irreversible inhibition of the proton pump.
ATP can't bind to the active sites, preventing release of hydrogen ions into gastric lumen.
Adverse effect.
The most frequent significant adverse effects occurring in at least 1% of patients include
Central nervous system,
headache 70%, dizziness 2%,
respiratory system,
upper respiratory infections 92%, cough 1%,
gastrointestinal system,
abdominal pain 5%, diarrhea 4%, nausea and vomiting 3%,
neuromuscular and skeletal system,
back pain 1%,
dermatology,
rash 2%.
dosage form.
Omeprazole is available at tablets and capsules in strength of 10, 20, 40, and some market 80 mg.
As a powder for intravenous injection.
Most oral omeprazole preparations are enteric-coated due to the rapid degradation of the drug in the acidic conditions of the stomach.
Thank you for listening.